Good morning, class. This is Dr. Pratibha KS, Head Department of Biotechnology at KLE SNC, presenting important questions from paper 8, that is Industrial Biotechnology. In this lecture talk, today I shall cover 2 mark, 5 mark, 10 mark and 1 mark questions. I hope you make use of today's list of important questions. I request you to invest your quality time and if possible try to answer all the given questions. If in case you are not getting answer to some of the questions, kindly reach to me through comment box. Okay? As usual, before going through today's lecture talk of important questions, let's have thought for the day. Education is not the amount of information that is put into the human brain. It must have man making and character making and also life building. Further, it is also part of assimilation of ideas, right? Then only by means of education, world can be changed. Think about this. Now, let me begin the list of important questions from industrial biotechnology. First, I am covering here 2 mark questions. Okay, that is first one, baffles, impellers, define natural and synthetic media, thermophiles, what are antifoaming agents, why synthetic media is also called a defined media, pasteurization, preparation of inoculum, list the fermented foods, rotary drum vacuum filter, what is biowort, what are hops used in beer production, what is the full form of PHA, aerator, what are bioreactors. Okay, these are some important questions. Next, moving on to 16th question. Agitator, foam control, single cell oil, name any 2 fermented products, expand USP and define, name any 2 types of fermenters, what is meant by fermentation media, what is fed batch fermentation, importance of agitators in fermenters, what is sparger, what is synthetic media. Next, moving on to 5 mark question. Explain in detail techniques used in DSP. Briefly explain industrial production of alcohol. Give a detailed account on characteristics of enzymes. Discuss industrial uses of enzymes. Discuss in detail production of vitamin B12. Explain in detail airlift fermenter. Write short note on process of aeration, process of agitation. What are the advantages of airlift fermenter? The next question, write short note on sterilization techniques. Okay, these are some important 5 marks questions. Now moving on to the 10 marks questions. In that first one, substantiate different types of fermenters with their applications. Explain different steps in production of beer. Give a detailed account on characteristics of enzymes. Briefly explain recovery of products in fermentation. What is fermenter? Explain typical fermenter with neat labeled diagram. Next question. Discuss the production of amylase. 7th question. Write a detailed note on fermenter and different control aspects of a typical fermenter. Explain in detail production of glutamic acid and citric acid. Explain in detail production of penicillin. Brief account of steroid biotransformation. So these are some important 10 mark questions. Next, further moving on to 1 mark questions. In that first one, name any one organism used in amylase production. Define biotransformation. Name the organism used for the production of glutamic acid. Define immobilized enzyme. What are HEPA filters? Mention the organism used for the commercial production of citric acid. List major steps involved in commercial production of wines. Define downstream processing. The aeration is mainly provided to organisms present in pH probe. Dear students, kindly try to answer by yourself 2 mark and 1 mark questions. If you find any difficulty in 5 marks, 10 marks, kindly reach to me through comment box. Further, moving on to 11th question. What is crude media? Define sterilization. What are different types of sterilization process used in industry? Students, kindly focus on all 1 mark and 2 mark questions. That is very useful during your Viva 2. Okay. What are molasses? Mention the different methods of immobilization of enzymes. Which organism used for amylase production? In which type of fermentation the microorganisms grow inside the medium? Which organism is used for ethanol production? Which organism is used for lactic acid production? Expand SCP. 21st question. What is dry heat sterilization? Differentiate between a difference between synthetic and non-synthetic media. What are impellers? What is immobilization of enzymes? What is the principle involved in working of autoclave? Mention the steps involved in DSP. This is for 1 mark, only mention steps. Molasses and cornstarch liquor are successfully used for. Expand HEPA, SMF, SSF, MSG. Which all radiations are used for sterilization? Next question, flocculation. Okay. Next here, malting. Who discovered penicillin? Saccharification, steroids. List industrial uses of enzymes. What is meant by submerged fermentation? Disk turbine. Which all radiations are used for sterilization? What is moist heat sterilization? Mashing. List some of the important microorganisms producing enzymes. Okay. So this is all about some of the very important questions which comes under industrial biotechnology. I hope I made myself clear to you about important questions of industrial biotechnology, whatever we have explained so far. Thank you everyone for investing your quality time. This is Dr. Pratibha KS signing out for the day. Stay tuned with me, stay safe, stay healthy. Thank you.